In industry, the proteins are mainly purified using a chromatographic method. There are other methods like differential centrifugation or dialysis, and electrophoresis can also be used, but it is always the chromatography that has been used in industrial capacity. So we will go through a few of them of this protein purification method. So firstly, the differential centrifugation. So the separation of the protein has been based on the molecular size. So the cell is need to be disrupted, as we have seen in the last lecture, and then the organelles released from the cell is coming to a fluid, which is also known as homogenate, and which is then filtered to remove any complete cells or large pieces of debris. And this homogenate undergoes a centrifugation at a different speed and centrifugal force. And each time the supernatant and the pellet of the organelles are formed, and supernatant is transferred to the another tube and then centrifuged at a higher speed. So it mainly depends on the centrifugation speeds and time, and where the size of the particles is important. So that in maybe lower centrifugation speed, the big particles can pellet it down. So we can use that supernatant for the next centrifugation and use a higher speed so that the smaller particles can be pelleted down. And this is the way its purification of protein has been done using a differential centrifugation. Dialysis is also a separation based on molecular size. So the concentrated proteins or the cell particles, mainly the organelles, has been taken into a dialysis membrane using a concentrated solution solvent. The concentrated solution has been actually coming out from the dialysis membrane according to the size. So the bigger particle, they actually stayed inside the membrane, and the smaller particles, they actually move out and mixing up with the solvent. So this is the way the separation of the particles has been done. So depending on the size of our protein, it can be in the concentrate, it can be in the solvent, or it can be inside the dialysis membrane. Next, as a gel filtration chromatography, which is also known as a size exclusion chromatography. So this is again based on the molecular size, and the smaller particles of the protein of interest is actually going inside or penetrate inside the matrix of the stationary phase, and the bigger particles, they actually passes through those matrices. So at the end of the chromatography, the bigger particles can be separated first, and then the smaller particles. So this is just following the basic concept of chromatography, where you need a mobile phase where the protein or where the product of interest is present, and you have to use a stationary phase, which is cellulose, I think the Cefadex, agarose can be used, and from there the solute or the mobile phase has been passing through. So in this size exclusion, cellulose has been used, agarose has been used. So this is again using the size of the molecule. So next is the ion exchange chromatography. Again, the separation of the protein has been done based on the ionic charge. So ion exchange chromatography is based on electrostatic interactions between the charged protein group and solid support material. That is the matrix in the column. So that is the stationary phase. So this matrix has the beads, which has the opposite charge of the protein of interest. And the affinity of the protein to the column is achieved by ionic ties with the bead. the different proteins are then separated from the column either by using a elution buffer of different pH or salt, or using a salt solution of different ionic strength. So the positively charged ion exchange matrices are called anion exchange matrices and absorb the negatively charged protein, while the matrices bound with the negatively charged group are known as cation exchange matrices, and they just absorb the positively charged protein. Next is affinity chromatography. So the principles of the affinity chromatography is that the sample is introduced into the column containing the matrix attached to protein specific ligand via a spacer arm. So that is, the stationary phase has a ligand that has a specific affinity to the protein of interest. So only the substance with affinity for the ligand are retained into the column, and other substances have no affinity, so they are just eluted out from the column. So the substance retained in the column then eluted out using the either changing the pH solution or the salt solution or organic solvent. So this is affinity chromatography. This is sort of a demonstration of using the affinity chromatography by the matrix, and this is a spacer arm with the affinity ligand, and it is specifically shown for the enzyme that the enzyme is found here, and the enzyme is attached to the affinity ligand and then separated out. For antibody, the ligand using an antibody ligand in the spacer arm, and it can be, and then it is attached with the antibody bound enzyme. So protein purification by immunoaffinity chromatography. So in this case, the cell has been disrupted. I mean, in all of them, the cell needs to be disrupted first, and the protein mixture then obtained by differential centrifugation. From there, we need to separate out the desired antibody, which has the immune affinity to the chromatography as shown in the last two slides. So that is more of the same as the affinity chromatography. Affinity purification of histidine tagged recombinant protein. So in this case, the protein of interest. So normally we use the DNA fragment that is expressed to the protein. So we can attach some histidine molecule at the end of the stop codon and just before the stop codon. So when the protein is expressed, the histidine molecule also expressed at the end of the protein molecule. So there is a polyhistidine tags are generated when the protein is expressed. So we actually put some histidine codon just before the stop codon of a DNA fragment, and that DNA fragment has been molecular cloned and grown into bacteria or any other host organism for protein expression. When we got the protein, so the protein of interest, and then at the end of that protein, we also have a polyhistidine group. So this polyhistidine group has been used to bind that protein and separate it out using a column. It is similar to affinity chromatography, and they are normally affinity for the his tag for transitional metals like nickel or cobalt attached to the matrix of the top. So the nickel or cobalt is actually attached to the stationary phase. So when the protein with the polyhistidine group has been passing through as a mobile phase to the stationary phase containing the nickel or cobalt, this nickel and cobalt can bind with the histidine group and separate the protein. So this is for this lecture, and I think in the last lecture we will go through some of the other purification methods, mainly the Western blot, and that's the last one. Thank you.